to talk about trust. Trust matters. It is the fundamental, essential glue that makes our society work. And in a way, it's the secret sauce with which individuals can come together and share in a project. As humans, we are fundamentally social creatures, and we actually like helping each other. And it's that very trust that powers that benevolence. So just imagine what it would be like if, for every decision that we need to take, we were to attach a bunch of prerequisites to it, and if we would doubt every motive. And actually, in a way, we would consider it even a sign of fundamental difficulty if we were consistently being asked to come up with confirmations of friendship, assurances of reciprocity, or just general proofs of goodwill, and nothing would ever get done. So we are who we are because we have this fundamental trust in people around us. And we extend that even to individuals who may not directly be known to us as individuals. So we generally do things in good faith. We are glad to do it, and we believe that others will do it too. There are no endeavors that really profit more from the occurrence of trust and the power of trust than the efforts to keep us healthy. So healthcare is an incredibly complex system that really requires that many parties will come together who often have inherently conflicting interests in that. And they need to come together with the main goal of getting this person healthy that is in front of them. So really, trust is the essence of that. And the more effective our healthcare system is, that is purely dependent on how much we can harness that human superpower of trust. So the patient-doctor relationship is at a central part of healthcare. And it really depends that the patient knows that the doctor is there truly to make them get better. The patient needs to open up with honesty and without judgment, and only that will then give the opportunity to have the correct diagnosis, to have the best care, and the most beneficial outcome. So it's all predicated on the existence of good communication and trust. Now, the doctor who is diagnosing or treating the patient is doing that while facing a whole set of different things that are unclear. So there are several things that are unclear. So the patient may present with a certain set of symptoms, and the doctor will have to pick which most likely condition will be at the root cause of these symptoms. There may be multiple different possible therapies that could be prescribed. The doctor will have to choose one of these, and there's some question about that. And then, of course, the individual treatment that the person will get will depend on, the success of that treatment will depend on the individual parameters of that person, the unique genetics and the unique physiology. So all of these are unknowns in this equation. What should not be unknown is that the doctor who prescribes these drugs would have any doubt in the fact that they are not safe and that they are not efficacious. So every country has their own regulatory framework for deciding which drugs are beneficial. And in the US, that task falls to the FDA. So one thing I want to get clear right at the upfront is the FDA does not have the political mandate or the funding to independently verify any claims about products that are placed in front of them. So the proof falls to the pharmaceutical company who has to present the data to the FDA And if the FDA review considers them beneficial and that the benefits outweigh the negatives, then that would be an approved drug. So the proof falls to the company to provide accurate and complete data to the FDA. So the FDA then asks the pharmaceutical company to first look at preclinical data, do tests in other animal models. If these work well, then the FDA will approve the drug to also be tested on humans in a set of clinical trials. Phase 1 of these clinical trials is giving the drug to healthy individuals to look at basic safety profiles. Phase 2 will start to give it to individuals with a particular condition to test efficacy of the drug. And then phase 3 will expand into a lot of different populations, different dosages, different interactions with other drugs. I almost forgot, and I knew I was close to it. I really wanted to give a shout out to EHR Data, who had the presentation before us, because the 4th phase of these clinical trials is, once the drug gets approved, there is the continued mandate to monitor safety of these drugs, because many of these long-term effects will only come out once larger numbers of people have taken the drug and they have taken them for long periods of time. So EHR is uniquely positioned to really capture that information and shine light on this very important component of safety for drugs. So in summary, the FDA doesn't do the testing itself. It relies on data from the pharmaceutical industry. It does have the opportunity to perform audits, to look at the places where the data came from. These are generally very intrusive affairs that will start to go through various quality management systems. They will look for compliance with different management practices in terms of manufacturing and laboratory, and so on. But the majority of the conclusions will be based on the data provided by the pharmaceutical industry. So this is where the quote that was attributed to the FDA director in Katherine Eban's TED Talk really shows the vulnerability that is faced in this situation. And I'm going to hand it off to Phil for this.
Thanks, Robert. Excuse me. The approval system requires the ethical behavior of the applicant. Otherwise, the whole house of cards will fall down. What does this mean exactly? It means that the drugs that we've been consuming globally since the beginning of time is all predicated on, as Katherine sums it up, the honor system. So who is Katherine Eban? Katherine is a New York Times best-selling author. She is a TED Talker, if that is a word, and she is an investigative journalist. And in 2008, she was contacted by an associate telling her about this slew of complaints that they were receiving from patients who had recently been switched from their brand name drugs over to generics. These complaints eventually funneled their way up to the FDA, where the FDA shrugged the whole thing off. They basically said, you know what? It's psychosomatic, the drugs look different, ergo, the consumer doesn't think they work. Now, Katherine and her associate, they were not buying this answer from them. And so she embarked to find out the truth. What was wrong with the drugs? Katherine's decade-long journey to answering that question lies in Bottle of Lies, where she shines a light on the FDA and the very shaky foundation in which they operate from to approve these types of pharmaceuticals. Things like, the applicant must be ethical. Great, we should all be ethical, right? But the data must be pristine, and the data must be shared at every single step. So early in Katherine's writing, she releases an article, and this catches the attention of an anonymous industry insider who refers to himself as $4 refill. $4 is apparently the cost to refill or get a generic drug at the Walmart. And so $4 refill goes on to tell her that if she really wanted to get to the crux of this issue, she needed to start to look where a majority of our drugs are made. What kind of majority? 80%. 80% of the active ingredients in all of our drugs, not just generics, not just name brands, all of them originate from these countries. One of the companies which were investigated, an Indian company, excuse me, was Ranbaxy. Ranbaxy at one time was ranked the 8th in the world as far as production of generic drugs. And thanks to a whistleblower, investigators were able to learn or discover that data from over 200 products that were operating or currently being distributed in 40 plus countries, including here in the US, had contained falsified data. They were substituting lower cost, cheaper ingredients. They were just straight up creating reports out of the blue, 3, 6, 9, 12, 18-month stability studies, all in the span of an afternoon. Ranbaxy, who was the applicant, was not ethical. And of course, their data was not pristine. Now, after an 8-year investigation, Ranbaxy eventually pled guilty to 7 felonies because of this fraud. They amassed about $500 million in fines and settlements, and that was it, right? The good guys won, the bad guys lost. Unfortunately, yes and no. What ended up happening is that Ranbaxy was really just the tip of the iceberg. They weren't an outlier. They were part of a system that has been around for a long time. And as it turns out, the trust that we've extended to these governing and regulating bodies, those who are there really that we give them trust to keep us safe. Well, because of things like relying on trust and relying on somebody to be ethical, this trust has been abused habitually for decades. So Eban had discovered more gruesome details. While Ranbaxy was certainly the biggest fish to fry, they were, again, by no means were they the only person who was doing this. During an investigation of 86 plants, 67 in India and China were found to be actively engaging in fraud, doing the same things that Ranbaxy was doing. Similar to how sweatshops exist to make cheap clothes, the same thing existed for the pharmaceutical industry. As part of that, Eban also had discovered some really gruesome details, things like dual-track production. And this is when those who have money get cleaner, safer drugs, and those who don't get drugs that actually fail trials. They're distributing failed drugs into poorly regulated markets like Africa, Southeast Asia, and in India. This problem, has gotten so bad that public health researchers have been able to directly link a rise in drug-resistant infections to the antibiotics that are being produced from these regions. And let's be honest here, the US, we're no gem either. Oftentimes, the FDA's public health mission directly clashes with that of a political mission, which is to have a consistent, steady stream of cheap drugs. These cheap drugs are what we get from the Affordable Care Act. It's the first line of fire. Medicare, Medicaid, all rely upon these. And in turn, we also end up consuming drugs that have toxic impurities and are just not bioequivalent to what it is we're supposed to be getting. Jumping the gun here. So how does all of this happen? Well, for starters, here in the United States, the FDA can walk into any facility unannounced and do an inspection. Whereas overseas, that's not the case. Instead, the FDA and regulators, they work hand in hand with these facilities. They schedule airport pickups and drop-offs. They help them get their hotels in order. This provides these companies an opportunity to have a massive runway to create, as she calls it, a facade of compliance. So our system that is built on trust and assumptions of ethical behavior is a failed one. There are far too few checks and balances that often are not engaged until too late in the process. So how do we fix all of this? Well, Katherine goes on to suggest, and the Veridat team here, we completely agree that the best way to fix this problem is going to be through rigorous oversight. And by rigorous oversight, it means that we should be at the absolute least verifying the truthfulness of the tests that are run, when they are run, and the data that is produced from them. Now, while Katherine falls short of discussing what an application of rigorous oversight could look like, We had a pretty good idea. And so we opted to pick up the ball and run with it. And this is where Veridat and our flagship service Bench comes into play. In a workflow or a regulatory body where Veridat and Bench exists, regulators can be equipped with the absolute confidence that they require to make decisions about a drug's validity, and consumers have the confidence that the drugs they take have emerged based on accurate and verifiable details. So what exactly is Veridat? Simply put, Veridat is a SaaS platform that makes it easy to capture critical, time-dependent information to the BSV blockchain. Why BSV? If you're watching right now, I hope you already know the answer to that question, but if you don't, we'll do this again for the people in the back. BSV is the only public blockchain that exists that can support the transaction cost, speed, and scale required to service enterprise-sized behemoths like the pharmaceutical industry. With that said, Veridat is a workhorse. It's a canvas. It's a place from which we can build several tools and services that will support a myriad of industries and services. But for right now today, we're talking about mitigating fraud. We're talking about drug development and safety, and really we're talking about saving people's lives by way of making sure that they're getting the drugs that they're supposed to be getting. Bench was born out of this desire to burn down this honor system, to solidify research integrity and data authenticity by capturing that information to the BSV blockchain. So because of the volume and variety of disparate systems that exist within the pharmaceutical testing and manufacturing arena, we knew that Bench had to be lightweight. It needed to be easily and universally implementable. And all of the services interacting with and supporting BSV needed to be invisible. We couldn't have the end user establishing wallets, managing private and public keys, buying and storing BSV. It wasn't gonna happen. Again, it had to be invisible. So with that in mind, we created a simple but powerful RESTful API that allows the end user a significant amount of flexibility to implement where in their workflow the key points of data need to be captured, hashed, and stored on both BSV and within our cloud-based system. And through a web-based UI, iterations on data files can be easily retrieved through search, sorting, and filtering options, which streamline report generation for audit processes and shows the data's trajectory files from the first save to the last, or as we call it, ironclad version control. So what does the standard workflow look like? Once the user defines the data points within their workflow, they will make a call to our API every time these points generate data. We then store that data, we create a hash of it, and that data then gets posted to BSV. Once the transaction completes, we amend that record with a transaction ID as well as the block ID. Subsequent edits of that record within the user system will make another call to the API. Again, the data is stored, another hash is created, it goes up to BSV, the block ID comes back down, and then we append the record also with that original transaction ID. So it doesn't matter how often you muck with the data, we'll always know what that original starting point was and where you ended off. This will be especially important for those who may find themselves on the business end of an audit. And now I would like to have Robert come back out here and discuss his utilization of JuvaTech at his innovative startup, JuvaTech.
Thanks, Phil. So I would like to give you some indication of how we've been implementing Bench into our workflow. So JuvaTech is a contract research organization for the pharmaceutical industry who's trying to insert during the preclinical testing using a novel animal model system to get some high throughput information about elements that are usually very difficult to get. So normally lead development at a pharmaceutical company uses a lot of methods that can be really scaled to massive levels of throughput. These include all kind of binding assays, anything that you can do imaging in a petri dish, cell culture work. And so all of this can be done at massive levels. The part that still remains expensive, frustrating, and extremely low yield has been for the longest time the behavioral testing in whole organisms. This part is actually very important because this is the main reason, that the main information that would give you some indication of why a drug may fail later on, either because it identifies some side effects that you really can't manage, or because it just lacks the efficacy. So we figured at JuvaTech, if we can bring down the price point of doing behavioral testing in animals, then that would give us an opportunity to insert that type of information much earlier in the drug development process, and really give it much more of a central stage, rather than only using it for the final validation, because all the testing is currently be done in rats, mice, dogs, horses, and the towering cost of that really prevents us from using that in the earlier stages of drug development. So what we did was we built our behavioral testing. We really started over with trying to bring behavioral testing to high throughput methods. So our automated computing instances will capture massive amounts of behavioral data that would be equivalent to, otherwise they would require whole armies of human observers sitting there with clipboards. As a second side, we decided to adopt a suggestion from Nobel laureate Eric Wischhaus, who suggested that we should consider fruit flies for this. And it doesn't take a huge leap of faith that we can test many fruit flies for the same cost that it would take you to test one horse. But really, the reason for us using fruit flies goes much deeper than that. There's a huge amount of common ancestry between the 15,500 genes flies have and the 20,000 genes humans have. Actually, about 75% of all human disease genes that we know are linked to some human disease have a direct and functional equivalent in flies. There are incredibly powerful genetic engineering tools where we can turn on genes, turn off genes, make them inactive, and we can create them into stable genetic lines that we can then cross with each other and make higher order combinations of everything that we want. So because we've been doing this at pretty massive scale, we're also creating massive metadata. And that from the beginning became very clear that we needed to handle that with some electronic solutions. So I want to quickly give you some idea of what the normal flow of a drug development is, and attach some approximate costs to this. So these are what our estimated out-of-pocket costs are. The drug company initially starts with lead development, where they decide on some kind of physiological process or some kind of molecular mechanism that is of interest to them. Then they build some molecules that specifically are designed to fit that mechanism and interfere with it. The thing that I would like to point out, that the rate at which a drug that has gone through lead development, that it will actually reach the market is very small. 1 in 10,000 of these drugs will actually make it all the way through. So most of these drugs that get chosen will go into preclinical development. And at preclinical development, most of these drugs will fail because it turns out once you test them in whole animals, there are side effects, toxicities, lots of other aspects that you would have missed at the initial stages in binding assays and cell culture assays. So the point where JuvaTech inserts is just before the preclinical data, where we, for a very small fraction of the cost that preclinical data will take, we can give some early indications of what the probabilities are that there are some serious issues or some positive data associated with that type of pharmaceutical approach. So we were thinking about that the way to explain that to a gambling crowd, for example, would be if you sit at a roulette table and somebody gives you some insight for a small fee, whether there's an 80% chance that the next ball is going to land on red, I assume that you probably would be quite excited about getting that information. The only other thing I want to point out in this slide is that basically, even after preclinical trials, the probability that the drug will reach market can be very low. And that is true for especially drugs that interfere with diseases and conditions where the pathogenesis is very poorly understood. So for example, neurological disorders with any kind of, for Alzheimer's, for example, have a very poor hit rate. Only 1 in 100, even after they pass preclinical work, will make it to FDA approval. I'm going to finish up with just giving you a very quick idea of how we inserted Veridat into our workflow. You'll recognize the left side of the story as a standard research workflow. We generate behavioral data in a paradigm. In this case, this is a paradigm where we can measure food consumption of single flies. We've got a computing instance that will generate phenotypes from that and write that to a network-attached server. This is very standard. What we then do is we interfere with, intercept at this point some of the metadata, write them out to Veridat. There was no change to our workflow required. All we needed to do was write the metadata out to Veridat. Veridat does the elements that Phil already described. At the end of the run, we calculate another hash that describes the content of the data file that was just generated, and we fire that off to Veridat as well. It gets linked to the metadata, and from that we can generate a complete record of that event, of that experiment that has happened. I will skip this and just go to the last slide to make the point that if we have a way of verifying information, we can actually bolster the value of trust. Trust will be given more easily and more readily if we have an opportunity to verify that the information that we're trusting is actually correct.